My name is Ophelia Ong, and this is my video assignment titled Genome-wide Association Studies, GWAS, and Other Multiomic-Based Approaches in Combating COVID-19. We are at war, declared Dr. Tedros, Director General of the World Health Organization, in March 2020. A few months prior, the world would not have known that this war was not between countries or militaries, but between humanity and the virus SARS-CoV-2. The first case of SARS-CoV-2 infection was detected in December 2019 in Wuhan, China. Within a few months, the virus spread like wildfire to other countries. In March, WHO declared COVID-19 as a global pandemic. If you know the enemy and know yourself, you need not fear the result of 100 battles. According to Sun Tzu, in this battle with COVID-19, it is important that we know the enemy, SARS-CoV-2, but also ourselves, the human host. Thus far, we know that SARS-CoV-2 spreads primarily through respiratory droplets to enter the respiratory system, causing flu-like symptoms such as cough, fever, muscle aches, and other symptoms. However, variation in severity of symptoms are seen. Little is known on why some infected individuals develop mild to no symptoms, while others have severe symptoms such as respiratory failure. The cause of these variation in clinical manifestations may lie in our genes, and the answer could possibly be found through the use of GWAS. Genome-wide Association Studies, GWAS for short, is a tool used to identify single nucleotide polymorphisms, also known as SNPs, and associate them with predisposition or resistance of individuals to certain diseases. GWAS is a tool to decipher ourselves, and in this battle with COVID-19, the weapon scientists are using to fight against this pandemic. Through previous studies, we know that certain groups are more susceptible to infection, such as the elderly. Statistics also show that males have a higher mortality rate when infected, and the presence of pre-existing diseases such as cancer, diabetes mellitus, hypertension, and other diseases also increase the risk of COVID-19 significantly. Currently, there are no effective vaccine or treatments available. Only supportive care is given through supplemental oxygen and mechanical ventilation when needed. Remdesivir, chloroquine, and hydroxychloroquine and other viral drugs have also been used in an attempt to treat COVID-19. However, none have shown promising effects in large-scale studies. Previous GWAS studies on COVID-19 by Ellinghaus et al. 2020 found two genes linked to respiratory failure. The first gene was located on a loci that encodes blood type. It was discovered that blood type O was the least susceptible to infection, while blood type A was most susceptible to infection by COVID-19. The other associated gene is a multi-gene cluster located on chromosome 3. For this study to be conducted, there are several ethical considerations. Firstly, the privacy of participants must be protected. Secondly, researchers should give feedback on findings to participants unless stated otherwise. Lastly, the study must also adhere to ethical conduct set by the International Working Group on Ethics. The overview of the methodology of this study is shown. Firstly, the case and control is determined. Second, the samples are collected and processed. Whole genome genotyping is then performed using the Illumina platform. The resulting data is required to go through several quality control steps to prevent type 1 and type 2 error. Lastly, GWAS is conducted and the resulting data is shown in a Manhattan plot. All participants should be in the age range of 18 to 40 and have no pre-existing medical conditions. The case will be participants that have tested positive for SARS-CoV-2 using RT-PCR and have experienced severe symptoms indicated by the need for mechanical ventilator or supplementary oxygen. For the control group, participants must be tested positive for SARS-CoV-2 using RT-PCR and were asymptomatic throughout the course of infection. Blood of participants are collected, and from the Buffy code, DNA extraction is performed using a DNA extraction kit. Whole genome genotyping is performed using Illumina Human HAP 550 platform, and Plink is chosen for the management of data, quality control, and statistical analysis for this study. The first quality control to be performed is to evaluate the sample quality. Sex inconsistencies and chromosomal anomalies is checked to detect sample identity problems caused by errors in sample handling. Sample relatedness is also performed to estimate pairwise kinship between participants. Population stratification is also performed to avoid bias and ensure samples are selected from a relatively homogeneous population. Removal of samples with low genotyping efficiency or call rate is performed. Samples with high failure rate in SNP assays indicate poor sample quality and should be removed. The second QC evaluates for batch effects. This is done by calculating average minor allele frequency (MAF) and average genotyping call rate across every SNP for each plate. Differences between average MAF and call rate between plates are determined to ensure no batch effect has occurred. The last QC is to determine marker quality. Marker call rate is evaluated and SNPs which fail on a large number of samples are removed. Rare SNPs with MAF of less than 0.05 is also removed. The SNPs are also checked for adherence to the Hardy-Weinberg equilibrium, and SNPs that are out of the Hardy-Weinberg equilibrium will not be eliminated, but they are flagged for further analysis during GWAS. The association test is performed using a single locus association analysis. A p-value of less than 0.05 indicates that an association is significant. Replication of studies with the same independent sample set is also required to validate the association. The data obtained from both primary and replicate GWAS will be displayed on a Manhattan plot of negative log 10 p-value. The threshold is set to a standard 5 times 10 to the power of negative 8. The resulting Manhattan plot should look like the following figure, where high p indicates association with a phenotype. Our ability to read out this sequence of our genome has the makings of a philosophical paradox. Can an intelligent being comprehend the instructions to make itself? We are a long way from truly understanding the instruction to the creation of ourselves, and GWAS only gives us a glimpse and small hints of these instructions and how they affect the outcome of diseases. When used correctly, GWAS is a powerful tool to identify the role of gene variation in COVID-19, and resulting data could potentially change the outcome of this pandemic.